Good morning, everyone. We'll be starting in 2 minutes. A very warm good morning to one and all present here. I, on behalf of ICIBLS 2020 committee, welcomes you all to the day 3 of the conference. Today, we have a great session lined up, starting with Dr. Kajal Biswas and Dr. Manchester Sen Gupta from NIH, Dr. Sushil Kumar Jha and Dr. Tulika Prasad from JNU, and Dr. Umesh Kumar from IMS Ghaziabad. Along with this, we have a panel discussion after the lunch. So with this introduction, I would like to kickstart the first session of the day. We have with us Dr. Kajal Biswas. He is a staff scientist at National Cancer Institute, NIH, USA. He did his MSc in biotechnology with Madurai Kamaraj College, University, India, and achieved his doctorate in biology from Julius Maximilians University. He was then accepted as postdoctoral research fellow at National Cancer Institute, NIH, Maryland, and got later posted as project scientist at Department of Microbiology and Howard Hughes Medical Institute, University of California. He was awarded the Fellow Award for Research Excellence from National Institute of Health in year 2010 and Federal Transfer Award in the same year from Center of Cancer Research. He has immense scientific aptitude, and also he has authored numerous papers in high impact journals. He will be shining the light on Today, he will be shining the light on the management of hereditary cancer by targeting BRCAness and functional classification of inherited mutations. We are glad to host you, sir. Over to you, sir.
Good morning, everyone. So, can you hear me?
Yes, sir.
Yeah. Okay, good morning, everyone. And first of all, I would like to thank Saksham for the nice introduction and also the organizer of ICILBS for giving me the opportunity to present my work. And it's really an honor to present my work in this platform. So, and I would also like to thank the convener Ashmita Das for inviting me for that. So I'd like to start the sharing the screen. So just a moment. Okay, is it visible?
Yes, sir.
Okay. So there is a minor change in the topic of this presentation, which I sent it to you. So I think, but the overall, it's the same on the one. So I would talk today about this understanding the survival mechanism of BRCA2 null cells or the BRCA2 null cells to target BRCA2 deficient cancers. So I'll start with the overall introduction that there are the two types of genes which are involved into the development of the cancer. So one is the oncogene, and in this case, in the oncogene, it's the activating mutation which activates one of the gene or more genes so that the cells undergo uncontrolled division. And in case of the oncogene, it's just one hit is necessary to just activate the gene and go into the uncontrolled cell division. And the second category is the tumor suppressor gene. And in this case of the tumor suppressor gene, the normal cells, we have the two copies of any gene or the two alleles. And then when the first mutation happens, the first allele get mutated, that the second functional copy take over the function. But when the second allele also got mutated, it goes into the uncontrolled division. So in case of the tumor suppressor genes, the two hit is necessary to form the tumor. So here today, I'll mainly talk about this tumor suppressor gene, about the BRCA2, which is a tumor suppressor gene. And the mechanism in the formation of the tumor suppressor gene to develop the cancer is two types. Like there are the caretaker's tumor suppressor gene and the gatekeeper tumor suppressor gene. So in case of the caretaker genes, they maintain the overall genetic stability like the DNA repair genes. Whereas the gatekeeper genes, they directly suppresses the growth, like the cell cycle regulator genes, the checkpoint control genes, or the apoptosis related genes. The both of these kind of gene both these genes, like they suppress the mutations or the abnormal cellular behavior. But along with this, if the caretakers are like the DNA repair genes, if they get mutated, the abnormal cellular behavior is still suppressed by the gatekeeper gene because they put another level of the control. So to take over the malignant phenotype by the caretaker gene, it is also needed the gatekeeper gene to get it mutated. Whereas the gatekeeper genes by itself also can develop the malignant phenotype. And we see that all the cancers, either by the oncogenic or the tumor suppressor genes, they are the genetic, like it is related to the genetic, but not actually hereditary. But there are certain percentage of the cancers are there which are hereditary. So what's the difference between the sporadic cancer tumor suppression and the hereditary cases? So in case of the sporadic tumor suppression, we have this normal cell which have the two functional copy. And when there is a first allele get mutated, it is still act as a normal. So it needs the suppression of the second allele also. The second mutations are needed, which then can accumulate the more mutations and can undergo the malignant cell. Whereas in case of the hereditary cancer tumor suppression, we inherit one copy of the gene which are defective. So we have only one functional copy in that case. So there are the more chances that another copy, if you get it mutated, the second mutation, then it can form the third and then malignant cell. So when you inherit one allele already non-functional, then those people have the more chances to develop the malignant cell. So what percentage of the cancers are actually hereditary? The majority, the 60 to 85% of the cancer is thought to be sporadic. And there are 10 to 30% is thought to be familial. So the familial are linked with a particular family, but they are not yet pinpointed to due to some gene. Whereas the 5 to 10% cancer is thought to be hereditary, and they are the which gene are involved in that hereditary cancer has been pinpointed. And there are a number of the genes has been identified in which are involved in hereditary cancer. And these are the list of the genes. And among these genes, like BRCA1 and the BRCA2 are our research interest. So I'll talk today mainly about the BRCA2. So when the BRCA2 cancer mutations you inherit, the lifetime risk of the developing the cancer increases. And it has been shown that the breast cancer or the ovarian cancer in females, it increases pretty high. Like the chances of developing the breast cancer is 50 to 85% of the people who inherit one defective allele of the BRCA2. Whereas the ovarian cancer, developing the ovarian cancer chances increases 10 to 20% if they inherit one copy of the defective BRCA2 allele. Whereas it increases also the male breast cancer, and there are also known that it increases the risk of prostate, laryngeal, and pancreatic cancers. But the actual magnitude is unknown. So what does BRCA2 do? So it is called as a molecular caretaker. So, and the tumors which are developed in heterozygote BRCA2 mutation carriers, that has been found that they lack the functional BRCA2. So when the tumors formed in those heterozygotes, they are functionally null for the BRCA2 gene. But the BRCA2 mutation carrier, the heterozygotes, they are normal. Whereas if you inherit both the copies of the BRCA2 which are defective or the partially functional, then those individuals, they develop a syndrome which is called Fanconi anemia and childhood cancer. And their maximum lifespan is up to 5 years. So they don't survive longer, and majority of, in case like BRCA2, if it is a null, they are not born yet. And what does this gene do? It is play a central role in DNA repair. So what, we'll give a brief overview with the DNA repair. So when a DNA, it is involved into a double strand break repair. So when both the strands of the DNA get damaged, either by radiation or due to some chemicals or during the replication process, so the double strand breaks are generated inside the cell. And when the double strand breaks are generated, there are two mechanisms which it can get repaired. Whereas the majority of the cells, it follows the mechanism which is called non-homologous end joining. So when the two strands are broken, and this, in case of the non-homologous end joining, it is just kind of tying a knot between the two ropes. So there are the number of proteins are involved here in these steps. And so it just join those two ends. And during this joining process, it can causes some deletion or addition of nucleotides. So this mechanism of the DNA repair is not very much error-free. So it is an error-prone mechanism, and it can generate the more mutations. But this is not cell cycle dependent. So it can happen any stages of the cell cycle. Whereas the other procedure, which is called the homologous recombination, and in this is an error-free mechanism to repair the double-strand break. And in this mechanism, it needs a template DNA to repair the DNA, that double-strand break. So the template DNA is available in the cells only in the S phase, or the synthesis phase, when the DNA has been duplicated before the division. So this step, this homologous recombination, takes place only during the S phase of the cell. And in that process, which is the error-free mechanism, there the BRCA2 is involved. So when the double-strand break is there, it is recognized by the proteins ATM, ATR, or the other proteins, and then it causes the chewing up of the one of the strand, which generates a overhang. And this single-stranded overhang of the DNA is then, with the help of BRCA2, is get coated by the protein RAD51. And then this nucleoprotein filament, it infects the homologous DNA or the homologous template DNA and synthesize the DNA using that homologous DNA as a template. And finally, DNA ligase ligate it up and repair this double-stranded break. So once this BRCA2 is found that it is involved into the development of the breast or ovarian cancer, so to study this gene function, the people started to make the BRCA2 knockout mice, and it has been found that when you make this BRCA2 knockout, the mice does not survive. And in case of the human also, we have seen that total BRCA2 null human was not found. Like they are not born. So this gene is essential for survival of the primary cells. And it has seen in our lab that when the BRCA2 heterozygous ES cells, they are surviving, but when you delete both copies of the BRCA2, there causes the growth arrest, or these cells do not survive. So this gives a BRCA2 paradox kind of thing. Like in case of the primary cells, you see that when both copies are mutated, they are going for the growth arrest. Whereas in case of the adult somatic cell, when both copies are mutated, they are viable and they form the tumor. So we think that there are the other mutations are taking place in these cells after the BRCA2 is non-functional, and those mutations help to survive these cells and causes the formation of tumor. So we wanted to see what are the other genes which help to survive this BRCA2 deficient cell. And thought a screening hypothesis that if we take this BRCA2 heterozygous embryonic stem cell, and if we mutate these cells first, and then we delete the functional copy of the BRCA2, and if some of the mutation which has been happened in these cells can help survive these BRCA2 null ES cells. And then we can identify those genes and see if they can actually play a role in formation of the tumor, or they can be used as a good target. And that's how we wanted to find a genetic interactors of BRCA2. And now this mutation can be of the two types. One can be, it can be activated gene, like it can be activated gene where the genes can get upregulated to help support for the viability of the BRCA2 null ES cells, or it might be needed a suppressor gene, like where you need to knock out both the allele of the gene and then you can get a viable BRCA2 null cells. So today I'll talk about mostly an activator gene which we found with a screen and how it functions. And to go into that, it is known that p53 is a functional interactor of BRCA. So when BRCA double is mutated, the double strand breaks are generated and loss of BRCA function. But if p53 protein is intact, they causes the cell cycle arrest and apoptosis. But if the p53 we delete it, it may cause the proliferation. And it has been found in many cancer or the tumor samples, the BRCA2 loss is associated with the p53 loss. But it's not all BRCA2 tumors loses the p53 gene. So we first wanted to test our embryonic stem cell that if the p53 deletion can support the viability of BRCA2 deficient mouse ES cells. So what we did, we took this, generated a conditional mouse embryonic stem cell where one copy of the BRCA2 is knocked out or made it non-functional, deleted, and another copy of BRCA2 made it conditional. So this was done by putting the two LOXP sites, which is a special DNA sequences on either side of the BRCA2 gene. And these sites, LOXP sites, are put about 80 kilobase apart, so that the whole BRCA2 gene, including whole the intron exons, can be taken out. And each of these LOXP sites is associated with a mini gene called HP or the RT. So when they delete this one, this mini gene come together and form a functional gene that gives to the resistance to the drug hat. Now we took this gene and put the recombinase CRE, which binds to this LOXP sites, and then delete the BRCA2. So if we delete this BRCA2 functional copy, so we'll get this HPRT functional gene, but there will be no BRCA2. So these cells we wanted, but when we delete in this conditional cells, we didn't find these cells do not survive. So, and this was confirmed by doing a sudden hybridization of each of the individual colony which we get, and we see that there are both the conditional BRCA2 allele and the mutated BRCA2 allele is present, whatever the colonies are there. So they are just a background colony. We didn't find any of the colonies or the cells which have lost the conditional BRCA2 allele and surviving. And next we took these cells and deleted the p53, both allele of the p53, and it was confirmed by the Western that there is no p53 gene expression. And then we took these cells and put the CRE here and delete the BRCA2, and we can see that there is a viable BRCA2 knockout over knockout ES cells we can get. And there are multiple cells we got where they have lost the conditional BRCA2. You can see here there is no BRCA2, conditional BRCA2 allele is present. And here, and these cells, due to loss of the BRCA2, we have seen that these cells, although they survive, but there is a huge growth defect. They grow slower, and that's why you see there is a fainter band of the mutated BRCA2 allele compared with the other cells. So it shows that the p53 deletion helped to survive this BRCA2 deficient ES cell. So we can use those cells for the screening. So to identify this activator gene, we took these cells and did a retroviral mediated random mutagenesis. And in this case, if we do this random mutagenesis, the LTR of this retrovirus, if it insert, it can activate the gene where it has been inserted, nearby gene. And then if we take this, after this random mutagenesis, the pool of the cells, and then delete the conditional copy of BRCA2, and see if we get any viable BRCA2 null mouse ES cells. And if we get this null ES cells, then we can find out that where this LTR has got integrated and which gene has been activated. And using this strategy, we found that one of the gene is called BRE, or breast and reproductive organ expressed gene, which can rescue this lethality of the BRCA2 deficient cells. And to confirm that this gene is, which we found into the screening, is really doing the work, so we overexpress this tagged gene in this conditional mouse embryonic stem cell. And we confirm that they are overexpressing this protein. And then we took these cells, did the Cree expression to delete this functional BRCA2. And we can find that there is a viable BRCA2 null ES cell. And you can see that we use the two independent clone, and in both cases, there are the survival of the BRCA2 null ES cells when there is a BRE overexpression is there. Whereas when there is no BRE overexpression, we don't find any of the surviving BRCA2 null ES cells. So it confirms that the BRE overexpression can help to survive the BRCA2 null cells. So how does BRE overexpression compensate that loss of BRCA2? So when the BRCA2 is lost, it is involved into the DNA repair. So there will be the DNA repair defect, and which will lead to the chromosome break or the genome instability. And once the cell accumulates the chromosome breakage or the genome instability, the cell cycle checkpoint function get activated so that they can get repaired and get it corrected. And if the cell cycle checkpoint function is intact, then the cell will stop the dividing. But if then the cell will not go for the division, and there will be a selective growth disadvantage. Whereas if the cell cycle checkpoint function is affected, then this genome breakage or the chromosome breakage will be tolerated, and the cells will undergo division with these genome errors. And there will be these cells get the growth advantage and can form finally the tumor. Now the thing is that what does this BRE overexpression do? Can it compensate this DNA repair defect of BRCA2 loss or not? So to test that, we took these cells which have been rescued by BRE, and you can see that in the heterozygote cell where the BRCA2 functional copy is present, when there is a DNA breaks, and this DNA breaks is marked by the presence of this gamma H2X protein. So when this DNA is broken, the double strand breaks happen, this protein gets phosphorylated and accumulates into this break site. And once this break site, it is there, and then the RAD51 protein, which is recruited by the BRCA2, it gets localized in that DNA damaged site. So you see that when there is a BRCA2 is functional there, the RAD51 gets recruited. But in case of this BRCA2 null cells and the MSCV BRE or the BRE overexpressed cell, we see the breaks are there, but the RAD51 protein is not recruited to repair the DNA. And when we quantify this, there is almost no of the RAD51 protein in there. So it shows that they are not compensating the function of the BRCA2, and this is also reflected by the karyotype of these cells. When we karyotype these cells, these BRCA2 null cells, they have a lot of chromosomal abnormalities, including the breaks or the fusion of these two chromosomes. And when we quantify all these different kinds of the breaks, you can see that there are a lot of accumulation of the genome alteration. So it shows that the BRE overexpression is not compensating the BRCA2 loss. So the next question is then that does it affect the cell cycle checkpoint function so that the cells are tolerating this chromosome break and it undergo the growth advantage with the broken cells, broken genome. So when that DNA damage responds, how it happens on the cell cycle, when the DNA damage is there are multiple proteins are involved into these cases. This is a short cartoon. Like when there is a DNA damage is there, the ATM or ATR kinases, they sense this DNA damage and get it activated. And then it gives the signal, and there is a lot of signal transduction process. And finally, it causes the activation of these proteins, CDC25A or P53, and which finally regulates the CDK or the cyclin-dependent kinases, and which acts at the different stages of the cell cycle. So we have seen that when the BRE is overexpressed, so first in these cells, we tested when we give in these cells the ionizing radiation or the radiation which will induce the chromosomal break. And if the chromosomal break is induced, the cells should stop the DNA synthesis to repair that. So we tested these cells. We took with the different doses of the ionizing radiation to induce the break, and see when in this graph, lower graph, you can see that when the BRCA2 heterozygous cells, or the one copy of the BRCA2 functional allele is present, with the increasing dose of the ionizing radiation, they stops the DNA synthesis, so that they are blocking the cell cycle so that they can get repaired and get it done so that the genome is maintained nicely before it undergo for the cell division. But in case of the BRE overexpressed cell, we see that after the BRE overexpression, even with the ionizing radiation, they are still going more of the DNA synthesis. So these cells, there are some defect into the cell cycle so that they are not stopping the cell cycle even with the DNA damage. And this radio-resistant DNA synthesis is a characteristic phenomena for a cells where CDC25A protein gets deregulated. So what does CDC25A does is that CDC25A is a phosphatase. So it removes the phosphate group from CDK. And then when the CDK is phosphorylated, they are inactive. But once this CDC25A removes this phosphate group from that, they turn into an active molecule and causes the cell cycle progression. So CDC25A helps to progress the cell cycle by removing this phosphate group. So what happens when there is a DNA damage is there, cell sense the DNA damage, CDC25A undergo ubiquitination and causes the degradation. So if CDC25A degrades, so there will be the less CDK protein and the cell cycle will be stopped. So we checked in these cells what happened with CDK and the CDC25A. So you see this in the left side, 4 row. You can see that with the increasing doses of the ionizing radiation, that CDC25A level is going down. So they are undergo degradation, and then phosphorylated CDK1 is increases. Whereas in case of this BRE overexpressed cell, the CDC25A protein level remains unaltered. So they don't get lowered down, and also the phospho-CDK1 protein is unaltered. And that helps these cells to maintain the CDC25A level and go into the cell cycle along with the DNA damage. Now, the question is that how BRE overexpression deregulates CDC25A after DNA damage. So we found that in this case, that DNA damage, when there is a DNA damage, there is a ternary complex forms between the BRE, CDC25A, and a D-ubiquitinase, which is known as the USP7. So the USP7 removes the ubiquitin molecule from CDC25A and then helps them to stabilize. So, and this was shown the co-IP experiment where we can see that all this interaction between these three proteins. So finally, we concluded a model from these experiments that when there is a normal level of this BRE and then DNA damage takes place, there is the accumulation of the DNA damage happens. And this accumulation of the DNA damage then activates a ubiquitin ligase, which ubiquitinates CDC25A. And then once it gets ubiquitinated, it undergoes proteasomal degradation and causes the cell cycle arrest and apoptosis. Whereas when there is a higher BRE level, in presence of the DNA damage, this BRE and USP7 forms a complex, and this complex is more formation. And then this equilibration between this ubiquitination and de-ubiquitination is shifted towards the de-ubiquitination of CDC25A. And there are the more CDC25A is present, which helps the cells to undergo the cell division with damaged DNA. So there is the viable cell with damaged DNA, which causes the accumulation of the more and more mutation, and that can form the tumor. Now to see if this is the case is happening, then if we overexpress the CDC25A, that should support the growth of the BRCA2 null ES cells. So to test that, we overexpressed CDC25A in this conditional mouse embryonic stem cell. And we checked that these cells are overexpressing the CDC25A. And then we put this Cree recombinase to delete this functional copy of the BRCA2 and see if we can get a viable BRCA2 null ES cell. And we see that after the CDC25A overexpression, these cells survive, that there is a BRCA2 null mouse embryonic stem cells are present. So it supports that the overexpression of the CDC25A can help the BRCA2 negative cell survival. And to test next that if the CDC25A expression is higher in the BRCA2 negative tumors or not. So we took the two different data sets available in the TCGA website, and we analyzed them for the CDC25A mRNA expression. And you see in both cases, when there is a BRCA1 or 2 mutation, those tumor samples have higher CDC25A expression. And this is a significantly higher, which supports also that there is a relation between the CDC25A overexpression and the BRCA2 loss. And next we confirmed with the immunohistochemistry how this BRE and CDC25A are expressed in this BRCA2 deficient human breast cancer tissues. So we took this BRCA2 null human breast cancer tissues and stained with both BRE and CDC25A. And here you can see that where there is a high BRE expression, majority of this high BRE expression tissues, they show the high CDC25A expression also. And when there are the low BRE expression, the CDC25A expression is also low. So and then we tested that if we overexpress this BRE, if it supports the growth of BRCA2 negative cells in vivo. So what we did, we took this BRCA2 negative mouse mammary tumor cells, and then we overexpressed the BRE in those cells. And then we injected into the nude mice and find that tumor volume. And we can see measure the tumor volume over the range of days, and when there is overexpressed BRE cells, they are growing much faster compared with the BRE negative, BRE normal expressing cells. And then to see, we took these tumors and analyzed if they have the BRE overexpression or not, and we see that majority of the tumors still maintain this BRE expression. And to support our data also, we see that CDC25A, how this CDC25A is there. And in these cells, we see when there is a BRE overexpression, the majority of them has the CDC25A overexpression also. So this shows, this further confirms our model that when there is a higher BRE level and the DNA damage is present, there is a high molecule of the BRE USP7 complex is formed more, which shifts the CDC25A ubiquitination to de-ubiquitinate. And then the cells can survive with the help of this elevated level of CDC25A and can undergo into the cell cycle. Now to see that, so we identify here that CDC25A, BRE, and USP7, the action of these three different proteins can help to survive the BRCA2 negative ES cells or the BRCA2 negative cells. So if any of these molecules can be used as a drug target to kill the BRCA2 negative tumor cells. CDC25A is a general regulator of the cell, so it is very difficult to get a CDC25A targeted. So we looked for these ones and see that USP7, it's not only in BRCA2 negative cells, but many other cancers also, it gets activated. And there is a clinical trial is ongoing, where they are trying to inhibit the USP7 in the liver cancer cell and see if it can act. So we tried to use also this USP7 inhibition in the BRCA2 negative cells. So if this USP7 is involved into the survival of the BRCA2 negative cells, if we inhibit that, they will inhibit the growth of the cancer cells. So you can see that BRCA2 deficient human colorectal cancer cell line, we took this cell line, which are the isogenic cell line of the DLD1, and there is a BRCA2 knockdown of these cells. And then we knock down the USP7 using siRNA. In both these cells, and we see that when there is a DLD BRCA2 negative cells, we knock down the USP7, there is a significant growth defect of these cells compared with the BRCA2 positive cells. And this data is further confirmed into another cancer cell line, human ovarian cancer cell line, which is sensitive that USP7 knockdown. So in this case also, we can see that BRCA2 negative cells, there is when we knock down the USP7, there is a significant growth defect. Now to test that this is also with the USP7 inhibitors, we took the two different inhibitors, P5091 and this HPX4108, and we see this colorectal cancer cell line. Also, when we treat these cells with both these inhibitors, there is a significant growth. It inhibits the growth of this BRCA2 negative cells. And this was further tested with the other cell line, also this human ovarian cancer cell line. And in both cases, we see that these inhibitors can inhibit the growth of the BRCA2 negative cells. So overall, so the summary of our study, we can present that the overexpression of CDC25A can help survival of BRCA2 deficient cells, and the expression of D-ubiquitinase along with BRE lead to CDC25A overexpression. And if we suppress this USP7 D-ubiquitinase, leads to enhanced killing of BRCA2 deficient cells. And this part of this study has been published in Nature Communication, and it's still ongoing with this study, some part which I have shown. And finally, I would like to thank our PI, Dr. Sham Sharon, and all our colleagues who are involved in this project. And thank you and all for listening, and I'm open to any questions.
Thank you very much, sir. In case we have any questions, please drop in the Q&A section.
Okay.
I would like to first start with the question session. It was an excellent talk, and I really think it was one of the best talks that we've heard so far, and it was really nice. First, I have a question that, you're talking about how that can be used in therapeutics also, right? Right. So, how do you think in therapeutics it could be used?
So we identify some of the molecules, some of the genes which are involved into the survival of the BRCA2 cells. So if we can target those molecules, so it can be used. So that's what I showed it also in the last slides.
In the last slide, last slides you were talking about the USP inhibitors and USP, you were talking about some clinical trials also that people are doing with that. So do you think in that, I mean, like the inhibitor studies that you are showing, it is excellent in, in vitro studies, but how do you think it will be useful in actual human models? I mean,
Yeah, we are just doing this in vitro, so there is a long way to go before it goes into the market, or if you say like human trial, without the trials, we cannot say. So we are just testing into the cell line now. The next will be into the mouse model or the preclinical study.
So do you think,
and then it will go back.
So do you think it will be useful to have a combinatorial thing? You have
definitely like that's the thing is that, so when we use it with the other drugs, like I have not shown it, but we have tested it, like the olaparib, which is now used for treatment of the BRCA2 null cells, null tumors. So olaparib or the cisplatin, which are commonly used now into the clinic. So when you use that drugs and it is more sensitive, it goes for these cells when you use the USP7 inhibitor, this one. But this needs the more studies also.
I understand. It's an elegant work, very elegant work.
Thank you.
Thank you.
Any other questions? So it looks like we don't have any other questions.
Okay, thank you.
Thank you very much, sir, for this intellectual talk. It was great learning exposure for us in the dive cancer. We'll be starting in another 5 minutes. Our next session, we have Dr. Manjishta Sen Gupta with us. We'll be starting in next 5 minutes.